The approval and the subsequent administration of COVID-19 vaccines have given the world a sigh of relief. However, the emergence of escape variants is raising questions on the usefulness of currently approved COVID-19 vaccines. Likewise, the mutant UK, South African, Brazilian, and Indian strains of SARS-CoV-2 are increasingly being reported in various parts of the world. To our knowledge, a comparative analysis of the strain prevalent in Pakistan with the mutant strains is scarce. To compare and contrast the gene and protein sequences encoding spike protein of SARS-CoV-2 between the commonly reported varieties in Pakistan with the original Wuhan COVID-19 virus. Spike protein is chosen because it is the primary target of many vaccines.
For comparison of viral strains, we focused on two clinically relevant parts of SARS-CoV-2, receptor binding domain and N-terminal domain. NCBI database was searched for the relevant sequences, multiple sequence alignment and structure analysis were used to investigate genetic variants and their implications. We show the mutations of spike proteins in Pakistani population relevant to the Wuhan reference in terms of positions and distribution. Figure 1 here represents the domain structure of SARS-CoV-2 spike protein. Table 1 shows the comparison of protein mutations, while Table 2 shows the comparative analysis of the 44 genomic sequences reported in Pakistan thus far. The data serves as a reference to consult the presence of particular mutations in spike protein in Pakistani population relevant to the original Chinese variety. Broad analysis of Pakistani variety with mutant UK, South African, Brazilian and Indian strains of SARS-CoV-2 is underway to be published in future. The variations observed may render spike protein resistance to the current vaccines, while some mutations like D614G is thought to have reached the fixation around the world. The D614G mutation is thought to increase the infectivity, but not necessarily to increase the severity of the disease. D614G mutations promotes the efficient binding of spike protein with ACE2 receptor. We have not found the D614G mutation to be accompanied by other three mutations that have been reported previously, which include C241T in the 5' UTR regions, a silent mutation at C303T7 position in the genome, while P323L mutations which was found in RNA-dependent RNA polymerase. Variations in spike gene and protein give us the extent of viral evolution under the selective pressure of vaccine and global spread.
Overall, there is a need for targeting viral antigens other than the spike protein. We propose that multiple antigens should be targeted simultaneously. The polyclonal response in such cases can deter the emergence of escape strains.